Thanks everyone for joining us. There's another episode in our series of webinars through ACES, and it's a great pleasure to welcome Robert Forster, who's joining us from Dublin. Robert has a personal chair in physical chemistry within the School of Chemical Sciences at Dublin City University. He's the director of the National Centre for Sensor Research, in 2020 elected to the Royal Irish Academy. Robert's got an outstanding research track record. All the normal metrics will tell you that. But I can also tell you through personal experience that he has all the respect of his peers, around the world for his ability to really dive deep into the fundamental knowledge that's required to deliver innovative solutions to practical problems. And I think you'll see that through the presentation today, that the fundamental knowledge is important, but the translation is also important. So we're really looking forward to Robert's presentation today. And I'm just going to hand over to him and we'll dive straight into it. Thanks, Robert.
Thanks, Gordon. Thanks, Lauren, Talveen, Phil, and everyone else for the wonderful organization today. It really is a great pleasure to have the opportunity under these difficult circumstances to share some of the work that we'll be doing, particularly the work that we've been doing with Gordon and the crew and Jun down in Wollongong. And really today I'd like to talk a little bit about the bipolar electrochemistry, the wireless electrochemistry that we've been doing, and to talk a little bit about the fundamentals of bipolar electrochemistry. What are some of the issues? What might we achieve using this electrochemistry in which the working electrode is itself not connected directly to a potentiostat, but you're relying on the generation of an electric field, and that electric field induces potentials within this bipolar electrode. It acts as its own electrochemical cell, and you can drive oxidation and reduction reactions at either end of this bipolar electrode. And then, as Gordon was saying, to discuss some of the potential applications of this technology, the work that we've been doing with Jun and Gordon and Shunyan and Jilan down in Wollongong to take arrays of biological cells, for example, nerve cells, and to stimulate those electrochemically or electrically using the wireless methodology. And so here, what's particularly attractive about this system is the materials challenges, trying to make surfaces that are biocompatible, that the cells will grow on, and that is also capable of being stimulated electrochemically. And then to move on to some analytical applications, to look at how we can use wireless technology to actually detect low concentrations of biomarkers, for example, in the blood of cancer patients, and use the light that is generated as a direct measure of the concentration of those biomarkers. And along the way, we'll talk about some of the issues, for example, the size of the electrode and how that influences the potentials that one can generate. And for some applications, you can use very large electric fields of the order of 1,000 volts or so per centimeter. And for those kinds of fields, you can generate things like hydroxyl radicals that will actually attack and decompose, and in most circumstances even mineralize, convert to CO2, ammonia and water, organic pollutants. So really it's a little bit of a journey all the way from biocompatible surfaces right the way through to environmental applications and trying to do wastewater treatment through wireless electrochemistry. So let's have a quick chat about the difference between conventional and bipolar electrochemistry. Obviously, we are all well familiar with the concept that this is redox chemistry. In other words, that a reduction and oxidation are always coupled together. And so when we, for example, drive an oxidation reaction at our working electrode in a conventional electrochemical cell, over here at the auxiliary electrode, you will drive the opposite, the reduction reaction. We typically have a very high electrolyte concentration, and that causes the voltage that we apply to that working electrode to decay very rapidly into solution, so that if you were maybe 100 nanometers or so away from the electrode surface, there is in fact no electric field in that solution. the electric field collapses over the double layer structure very close to the electrode surface. In contrast, when we have a wireless or a bipolar, an open bipolar cell, what we're doing is we're using a pair of feeder electrodes, and that pair of feeder electrodes set up an electric field in the solution. And now if we place an electronic conductor in that solution, it assumes an equilibrium potential, but one end will have a potential that is different from the solution potential. We'll generate an oxidizing potential at one end of the electrode. We'll generate a reducing potential at the other end of the electrode. And now this bipolar electrode acts as its own electrochemical cell. One end we can drive oxidation reactions, the other end we can drive reduction reactions. So the electrons that are produced at one end of the electrode are harnessed and used to drive a reduction at the other end. But of course, implicit in that model, there are some very big differences between the conventional and the bipolar configuration. If we think about the concentration of electrolyte in a conventional cell, we'd like that maybe to be 0.1 molar, or maybe 20 to 50 times higher than the analyte of interest. In contrast, in the bipolar situation, we want that electrolyte concentration to be very low so that we get a significant electric field through the solution. That electric field is what induces the potential within the bipolar electrode and allows us to drive electron transfer reactions within that bipolar electrode that acts as its own electrochemical cell. So we deliberately want to have these intense electric fields in the wireless or the bipolar situation. There is no connection, no physical, no wired connection to the electrode in the bipolar or the wireless electrode configuration. And again, one of the important things is that most electrochemists will carefully position their electrodes, for example, trying to keep the auxiliary electrode an equal distance from all points on the working electrode surface. You don't want to put that auxiliary electrode off to the side. Why? Because you want a uniform potential distribution across the electrode in the case of a conventional electrochemical situation. In contrast, for the bipolar situation, we deliberately, intentionally have a potential gradient across the electrode, the bipolar electrode itself. So one end reducing, one end oxidizing. And now if you think about some of the experiments that many people in this audience will be interested in doing, for example, maybe electrodepositing a conducting polymer film onto that surface, you now have a gradient of potential across that electrode. The film will grow on one end and won't grow on the other. So you can begin to think about making some interesting films or film thicknesses across that electrode surface. Of course, because we have an electric field within this solution, we are going to see electrophoresis or migration of ions in this electric field. That is completely swamped by the high electrolyte concentration in a conventional cell, but because our electrolyte concentration is low in the bipolar experiment, ions are going to move in response to that electric field. And finally then, we have this ease of multiplexing. With conventional electrochemistry, you have to make individual electrodes, you have to make a wired connection back to a potentiostat, and you have to have separate potential or voltage control over that electrode. In contrast, with the bipolar situation, you can put an array of electrodes within the solution, and each of those will adopt a potential. One of the things we're very keen on doing with Gordon's group is to try and shape this potential distribution within the electrolyte. So obviously with 3D metal printing, for example, you can make 3D electrodes, and the question then is, can you use those electrodes to shape the electric field and not just have a simple linear gradient of the electric field, but can you actually shape it in interesting new ways? And of course, like all new technologies, there are going to be victims of this wireless technology, and indeed when you think about conventional electrochemists, some conventional electrochemists are very committed to their conventional systems. You think about people who've been in the field for quite a while. Here's Gordon sitting next to his conventional electrochemical cell, looking a little quizzical about what the future will bring with all this wireless technology. So first of all, I'd like to talk a little bit about our work with Junshen and Jilan, and particularly Shunyan down in Wollongong. And what the guys at Wollongong are doing is they are taking bipolar electrodes, functionalizing them with conducting polymers that are biocompatible, arraying biological cells, nerve cells on the surface, and then trying to electrically stimulate those cells. This is what the setup looks like. You can see that the conventional potentiostat is replaced by a power supply. That generates voltages that are delivered to these two feeder electrodes. And right here in the middle is the bipolar electrode itself. And once again, there is no electrical connection, no physical wire to that electrode, that bipolar electrode. And what we're doing is we're using the potentials generated by the feeder electrodes to induce a voltage within that, that electrode. So what Shunyan has done is to take fluorine doped tin oxide and electrochemically deposit a conducting polymer, for example, PANI, onto that surface, and then look at what happens when you induce a potential within that electrode. So if you have no potential on the two feeder electrodes that would sit either side of this bipolar electrode, you get a uniform potential distribution within the surface. And then when you do optical imaging of that surface, you get a uniform color across the surface, i.e. a uniform redox composition of the layer. If you then poise one of the feeder electrodes negative, if you poise one of the electrodes negative on this end, it induces a positive potential in this end of the bipolar electrode. You get oxidation of the conducting PANI polymer on this end, and on the other end, that is still in the reduced state. If you change the polarity, now rather than having this end negative and this end positive, you can swap the polarity. You can cause the other end of the bipolar electrode to become oxidized. So we can control which end of the bipolar film is actually oxidized. And then when you remove the feeder potentials, you again recover the state that you had at the beginning. And very interestingly, you can do this also in more complex, more biocompatible buffers like PBS. And again, you can induce this redox, this distribution of redox states across the PANI film within this bipolar electrochemical cell. More significantly, again, you can change the anion that's within the polypyrrole film to, for example, dextran sulfate, or you can even introduce proteins within that layer, for example, collagen. And these act as focal adhesion points for the binding of particular cell types onto the surface, PC12 nerve cells in Wollongong and cardiomyocytes here in Dublin, both electrically addressable cell lines. So a little bit more detail about how the system works. One of the key advantages of the bipolar or the wireless system is that you can multiplex. So you can have 1, 2, 3, 4 bipolar electrodes in parallel, and all of those bipolar electrodes will then assume a potential that is dictated by the electric field strength as controlled by the voltages that you apply to these outer feeder electrodes. And so Shunyan has used 3D printing, for example, to make these arrays of chambers into which it can insert her FTO that is coated with this biocompatible polymer, and onto that surface then the cells are seeded typically at 20,000 per centimeter squared, and now you can turn on the voltage and you can begin to electrically stimulate these cells, see do the cells remain alive, do they remain viable, and then to see can you, for example, get them to differentiate from the normal undifferentiated effectively cancer cells into nerve cells in which you have these neurites, and can you get interconnections between those neurites, and can you form an array of communicating nerve cells on this surface. So these are some of the results that Chunyan has gathered down at Wollongong. Here we have PC12 cells, so these are progenitor nerve cells arrayed on this polypyrrole dextran sulfate collagen matrix. The collagen is present so that the cells adhere onto that surface. And what you can see here with no electrical stimulation is that a good population of viable cells. This is a pico green staining on the surface, and also using things like calcine dye to see whether the cells are alive or dead. And the important thing here from this first image is that the cells will actually grow on the surface and remain viable and active. They will remain active on the surface, no bipolar stimulation, and when you stimulate them electrically using the wireless technology, then those cells remain alive, and in some circumstances you may even get more cells growing on the surface than before. And then what Chunyan does is she electrically stimulates those cells, typically using maybe 1 volt between the 2 feeder electrodes. That generates about a volt potential difference in the conducting polymer, and she'll deliver that 1 volt for an hour a day for 7 days. If the cells are left without stimulation for 7 days, you can see that there's still a significant population of viable cells on the surface. But very significantly, with the electrical stimulation, the wireless electrical stimulation, the cells are still alive, they're still actively growing on the surface, but very significantly, they have differentiated into nerve cells. They produce these neurites, and you're now beginning to get a network of connected cells on the surface due to the electrical stimulation. So really wonderful science, biocompatible surfaces that cells will grow on, that are electrically addressable so that you can electrically stimulate the cells, and now you can induce differentiation in these PC12 cells and make them grow as nerve cells. And you can see that in a little bit more detail here. You can see the growth of the cells only growing their neurites only under electrical stimulation. So very exciting work in which you can electrically stimulate and you can actually begin to drive key processes in biological cells using this wireless technology. So we're also, Chunyan very kindly came over to Dublin last summer and spent about 6 weeks with us there, training us on how to do these biological experiments. And what we're interested in doing is addressing another class of electrically addressable cells, that being cardiomyocytes. And here what we're looking at is the deposition again of conducting polymers, but incorporating fibronectin. These cardiomyocytes bind to a fibronectin decorated surface. But remember, we talked a little bit about the bipolar electrode, and that bipolar electrode gets placed in an electric field. One end becomes oxidizing, one end becomes reducing. And now if you put that electrode into a solution that contains pyrrole, you can begin to grow wirelessly a polypyrrole film on this bipolar electrode. And what you're looking at here is the underlying glass substrate, about 100 nanometer thick layer of fluorine doped tin oxide. And then on top of that, down at this end of the electrode, about 100 nanometer thick polypyrrole layer. On the other end of the electrode that would have remained at a reducing potential, you do not see polypyrrole deposition because we're not oxidizing the monomer and therefore driving the formation of the film. And what's interesting is that this end of the electrode, obviously we're looking at distance along the electrode here. This is about 1 centimeter when we're doing the deposition. but the potential is varying during the deposition across this surface. And if you look here, here's the transition between the non-polymerizing and the polymerizing zone. And in some sense, we're actually mapping the potential distribution along this electrode surface. What's really interesting is that this is actually quite sharp. You go from no deposition here to essentially Nernstian controlled or kinetically unlimited deposition here within something like about 300 nanometers. Quite a lot sharper than we'd imagined that it would be. So there's influences of mass transport as well as the potential distribution going on there. One of the things we showed you from Shunyan's data is that you can swap the polarity of the electrodes. One electrode that's initially positive and the other one negative, you can cross those over and switch the direction of the polarity. And that will then cause the film structure to change. So that's what we've done here. Just to confuse, this is the positive end, this is the negative end. But what we're doing is we're swapping during the deposition, we're pulsing back and forth. And what happens then is you get a combination of deposition kinetics and mass transport. And what you actually get are these bands of growth within the polymer film. So you go from a smooth wedge-shaped film here to one that contains bands in the pulse situation. So again, you can control the potential, you can control it dynamically on this bipolar electrode in time, and you can use that to deposit it and control the structure of the conducting polymer. So Shunyan trained up Fionn in our lab, and Fionn again has been able to grow cardiomyocytes on this surface. So this is a polypyrrole film that contains fibronectin. What you're looking at is the growth of these cardiomyocytes on that surface. And what's interesting is that you can do the bipolar stimulation, and now you see changes in the cell morphology as you do that bipolar stimulation. So the combination of the conducting polymer sitting on the FTO allows you to control the local potential, and that influences the growth of these cardiomyocytes. And what we're trying to do here is to use the ability to change the potentials dynamically in real time and actually try and get these cells to beat as a coherent mass of cardiomyocyte cells. The other aspect that we're interested in looking at, again with IPRI and Wollongong, is the whole concept of generating light. Conor Hogan, for example, over in Melbourne, has done some pioneering work on the development of new dyes and trying to generate light, generate luminescence, not by optical stimulation, but by controlling the potential and having, for example, a co-reactant in solution. And this is classical Roche electrochemistry. One of the, we talk about point of care testing now for the COVID-19 virus and for antibodies. Roche have a system on the market now that is based on electrochemiluminescence. But fundamentally what happens in our hands is we have a ruthenium trisbipy type dye. This is the one that we're looking at the moment. It has an amine functionality that we can bind to biological molecules, but fundamentally you oxidize the ruthenium 2 at an electrode surface, it generates ruthenium-3. You then bring in a co-reactant. The co-reactant reduces the 3+ to 2+, but it is in an electronically excited state, and now that relaxes by the emission of light. So the key features here, we apply a positive potential, we oxidize the ruthenium, and the ruthenium then emits light. And so working with Gordon and Stephen down at UOW, if you can get these guys to stop making very high-end jewelry for the jet set, you can get them to make these interesting arrays of three-dimensional electrodes in titanium. And then what we do is we use those as complex feeder electrode systems. For example, here, what you're looking at is where 2 of those pins are positive. They're the positive feeder electrodes and 2 are negative. And we're trying to do COMSOL modeling of the field distribution within that pair of electrodes. And you can see that you get not just the linear decay directly between, but you get these interesting dips and troughs of intensity in the potential distribution of the electric field strength, depending on the configuration of the electrodes. And one of the challenges in this space is the Gouy-Chapman-Stern model, for example, very successful at explaining double layer structure of conventional electrodes. In these experiments, we're working at a very dilute electrolyte concentration, and you rapidly run into one of the fundamental underlying assumptions with the GCS theory, namely that the charges are point charges. And what that does is it allows you to pack as many ions as you want to onto the electrode surface. And of course, in physical reality, that is not true, but the models use that assumption and cause some problems with the COMSOL model. But then what we can do is we can begin to image experimentally that field distribution. So again, in here, you can just about see them are the two negative feeder electrodes. These are the positively charged electrodes. These positive electrodes are now causing particles. These are 100 nanometer conducting diloaded particles. And if they encounter a positive electric field, then they become oxidized and can emit light. What we're doing here is we're leaving the shutter of the microscope open for a long time, and these trails are actually the movement of the particle over time in response to the electric field that we're generating, and the fact that they are positive potential applied to them, positive potential induced by the electric field. We can generate light, but we can also map the distribution and compare the experimental response with the theoretical response. We can go to three-dimensional cavity arrays, for example, using nanosphere lithography, and make these open cavity arrays. We can look at the light that's generated from these dyes within the cavities. And now you see something that's interesting. If you take the planar surface, you get a certain intensity of light. If you array 2000 nanometer cavities, you get a brighter emission, 1000 brighter still, 200 a brighter still. And then when we go to 600, you get an even brighter emission, but the optimum cavity size is about 800 nanometers. So there's an interesting message in terms of size. Smaller, 200 nanometers is not necessarily better. 800 nanometer actually represents the most intense emission. And these are corrected for the charge path that really reflect the efficiency of the ECL response. So what we can do then is we can build classical DNA sandwich assays on that surface. We can put capture strands down on the surface. We can bring in our targets. If the target is complementary to one of our capture strands, we can see binding of that target to the surface. Then we can bring in a probe strand that has our luminophore, our ECL active luminophore on the surface. And now when we use the bipolar, we turn on the electric field, that will oxidize that dye. When the co-reactant is present, we can now generate light. And that's shown here. We get a bright emission only when our target is present. And so with that kind of approach, we can detect microRNA, for example, that are associated with the neuroblastoma, a childhood cancer neuroblastoma. And we can do these measurements in blood serum, and we can achieve detection limits of detection of sort of low to mid nanomolar concentrations. And then we can go and look at real patients. Traditional approaches use qPCR to measure those concentrations, and we can compare those results with our bipolar ECL data. We don't get a perfect correlation. A perfect correlation would be a slope of 1 and an intercept of 0. Our slope is a little bit higher, but it's reasonably close to 1. And you can imagine if this data point was excluded, we get a slightly lower slope, closer to 1. And relative to the scale of the y-axis, the intercept is approximately 0. So we really do get quite close to a direct, unbiased correlation between qPCR and the bipolar ECL. One of the other aspects that we've looked at is to take nanoparticles, and remember that the field strength you need to induce a particular potential is directly proportional to the particle size. So if we go from a 1 centimeter electrode, we want to generate a potential of 1 volt. Let's say these are ruthenium dyes on the surface. We need 1 volt to oxidize that ruthenium. If the electrode is 1 centimeter long, we only need a field of 1 volt per centimeter. But if we want to go to the nanoscale, we want to use nanoparticles, gold nanoparticles decorated with antibodies so we can target them, decorated with ruthenium dyes so we can do this ECL emission. Now, if this is 100 nanometers, we're going to need 10 to the 5 volts per centimeter electric field strength. And of course, as everyone in this audience knows, if you expose cells to a 10 to the 5 volts per centimeter electric field, what happens is that they burst, they lyse, they disrupt. And so we're interested in developing strategies to get around this, to exploit the power of wireless electrochemistry. You could imagine if we could assemble an electrode in situ, perhaps within the brain for deep brain stimulation and control of Parkinson's tremor, that would be a great technology. And so the idea is that we use these particles, functionalize with antibodies that will selectively bind to a cell of interest. These could be cancer cells, or they could be specific cells within the brain. And now the concept is that these nanoparticles assemble an electrode in situ. And now the size of your electrode is not 1 nanoparticle, it is the entire conglomerate or aggregate of those particles. And if we get enough particles in there, we can get a conducting path if we're above the percolation threshold. And now we might build an electrode in situ that is many hundreds of micrometers in size. So we've done some work on that. What you're looking at here are MCF7 breast cancer cells decorated with 100 nanometer particles. We place those cells in electric field. That electric field is about 10 volts per centimeter. And now what you're looking at is the luminescence coming off the ruthenium dye that is on those nanoparticles. These are aggregates of nanoparticles that are coming together, whereby the 10 volts per centimeter that doesn't do the cells any damage, you can see that over here, is actually sufficient to induce a 1 volt oxidizing potential that allows the ruthenium to become reduced to the electronically excited state and emit light. So very briefly then, just tell you about our work on wastewater treatment using wireless technology. The key advantage here is that you can obviously apply as intense an electric field as you like to the wastewater sample. You want to deliberately decompose things within that sample, and you don't care about destroying cells. If there are pathogens in there and you destroy them, that would be a good thing. You're not trying to preserve delicate biological chemistry. So the whole concept here is that we introduce wastewater. We have a reactor. The reactor generates hydroxyl radicals. These have a tremendous oxidizing power, second only to fluorine. Those radicals will attack organic molecules, pathogens within the water, decompose it, and ideally mineralize it to carbon dioxide, water, or if it's a nitrogen-containing molecule, ammonia. What we're doing here is we're using boron-doped diamond particles. These particle sizes range from 5 to maybe 500 micron. And we characterize those, and we use these now as discrete bipolar electrodes within a wireless electrochemical cell. So one of the things we're interested in is what is the effect of the boron-doped diamond composition on the performance of the hydroxyl radical generation. So what we've been doing is we've been taking a platinum microelectrodes, for example, in this case, a 7 micron platinum electrode. We've been polishing those to a planar surface and then etching them back to create cavities that are roughly 7 microns in diameter and roughly 7 to 10 microns deep. And the idea is that we introduce a single boron doped diamond particle into this cavity so that we can do single boron doped diamond particle electrochemistry in this cavity. And then we're looking at the effect of the boron doping level on the electrochemical performance. What's interesting is that at the 5 by 10 to the 20, or the 2 by 10 to the 21, so this is 5, this is 20 by 10 to the 20 boron particles per centimeter cubed, what we see at about 1.5 volts is a large increase in the current that corresponds to hydroxyl radical production. So irrespective of the radical concentration, irrespective of the boron doping, we can generate lots of hydroxyl radicals. But remember that in a bipolar experiment, one end of the electrode is oxidizing, one end is reducing. And just like any electrochemical cell, if ox and red are not balanced, that will, the slow step will dictate the overall performance. So in our case, we have to have a reduction reaction. The reduction reaction we would like to have is oxygen. And at the low doping level, the oxygen reduction reaction is really quite poor at this low boron doping level. It is much better at a higher boron doping level. We get much better defined oxygen reduction. That's important because we do not want this ORR to limit the production of the hydroxyl radicals. So what we've been doing then is we've been designing initially simple cells. Here's the space for our 2 feeder electrodes, and then we put a suspension that we keep stirred of the boron doped particles in here. We apply a potential to the 2 feeder electrodes, and then we generate the hydroxyl radicals at the boron doped diamond particles. Those hydroxyl radicals then decompose the organic of interest. Later on then, we're hoping to persuade Stephen and Gordon to devise some of these intricate 3D electrodes that will allow us to shape and control the electric field within the reactor. So one of the compounds of interest is our pharmaceutical drugs and metabolites, a growing problem in municipal water. And what we're doing here is we're looking at this drug, Cardiomyocytes, and we're now changing the voltage on the feeder electrodes within that reactor. We're generating really rather intense electric fields of the order of 2,000 to 3,000 volts between the 2 electrodes. So we're dropping a couple of thousand volts between these 2 feeder electrodes. It's really important to realize that not all of that voltage drop is actually delivered to the boron doped diamond particles themselves. There will certainly be some loss. We talked about that exponential decay that can occur depending on the electrolyte concentration close to a conventional electrode. The same thing can happen here. But what you're looking at is mass spec data coming off the headspace for that reactor as a function of the electric field strength. What we're looking for, 44 grams per mole is CO2. And what you see is that in the presence of the Cardiomyocytes, at low field strengths, you don't get any carbon dioxide. And now when you approach about 2,500 volts, you suddenly see the appearance of carbon dioxide. We are generating hydroxyl radicals. We are decomposing the carbon within this Cardiomyocytes. We are generating CO2. And one of the convenient ways to monitor that, this technology throws up some interesting challenges from a sensor perspective. How do you monitor what's going on inside that reactor? You're generating hydroxyl radicals. Any sensor material in there will be decomposed by the hydroxyl radicals that you generate. So conventional spectroscopy is a good UV-Vis spectroscopy, is a good technique. And what you're looking at here is the initial UV-Vis. We're electrolyzing for an hour. We're electrolyzing for 2 hours. We're electrolyzing for 3 hours, 4 hours. And after about 3 hours, you can see a complete collapse in this band at about 275 nanometers. And what we can do then is we can sample that reaction solution, do HPLC, and confirm that there are no organic degraded products left. It has in fact actually been mineralized which is a very exciting development. We can change the nature of the feeder electrodes. We started using platinum, and then we went to stainless steel. You do see some slowing down of the overall reaction a little bit. It takes about another hour using stainless steel as the feeder electrodes. And the reason for that is the electric field distribution is a little bit different with the stainless steel mesh than with the platinum. So this is probably the most exciting slide of all, the conclusions. I think with working very closely with Junshen, Jilan, and Shunyan down in Wollongong, we really do have a very nice conducting polymer platform that cells can adhere to that preserves the electrical connection so that we can induce a potential within that electrode, have a transfer faithfully to the growing cells on the surface, and use that potential to stimulate the cells. Significantly, that wireless stimulation can cause cells to differentiate. It can grow neurites out of PC12 nerve cells. It can get cardiomyocyte cells to grow in an oriented way with the electric field, and we're trying to get those cells to beat as a coherent array rather than in a random fashion as discrete cells. We think we can begin to contemplate the possibility of self-assembling in situ within biosystems electrodes that we can address wirelessly and, for example, generate electrochemiluminescence. And then these boron-doped diamond particles seem to be quite efficient at generating hydroxyl radicals so that we can decompose water in a wireless way. And of course, the great advantage of that technology is that it's very much scalable, because you're now exploiting the multiplexing capability. Each one of those particles, the boron-doped diamond particles, becomes its own electrode. And so you can take an entire volume of water and electrochemically address the contents in there without having to transport everything to a conventional electrode surface. So thank you very much for your attention. To pay a huge thank you to the collaborators at University of Wollongong, to thank Shunyan for coming over to Dublin and for training Fionn so expertly in the production of these films and the stimulation of the cells, and of course to our funders. Thank you.
Thank you, Robert. On behalf of everyone who's dialed in, thank you. If people have got questions, you can just send them through the chat or raise your hand, as Lauren said before. I'll just kick off, Robert, in terms of that, the assembly of the nanoparticles into electrodes. Are you using the field to drive that assembly, and do they actually have to be in full contact in order to realize an electrode structure?
So the only targeting that's present there is the antibodies that are mobilized on the surface, and they recognize a specific cell receptor, protein cell, surface cell receptor on the target cell type. And then between healthy and normal and the cancer cells are done, you get good discrimination using those antibodies. But you probably could imagine using the electric field to actually deliberately, as you suggest, migrate those particles with a charge to the surface.
Mm. And do they have to be? Sorry, do they have?
The second part of your question, Gordon, absolutely yes. They must be in electrical contact. And to achieve that, what we do is we avoid covering the entire surface of the gold nanoparticle with either the dye or the antibody. So there are contact patches where you can achieve electrical connection, albeit very inefficiently, between those particles. But fundamentally, what we're looking for is a percolation pathway through that array of particles. So yes, you need a high density of the particles, and you need to carefully control the coverage of organic material, if you like, on the surface. And one of the big challenges is to avoid protein corona encapsulation of those particles before they get to the site that you're interested in. So there's quite a lot of chemistry still to be worked out.
Okay, great, thanks. I'll see if there's any other questions in a moment. I've got one on the chat here from Frankie Rawson at University of Nottingham. It's a two-part question, Robert. So first one is, how do you differentiate electrochemical effects, I guess, from electric field effects in the cell experiments?
I think that's a really good point, because as Frankie says, not only do you have the redox-induced chemistry in the underlying from the underlying coated electrode, you also have this electric field gradient across the surface. So I think that no is the straight answer. I think that is something that we need to decouple. In terms of the reactive oxygen species, we are not deliberately generating ROS in those biological experiments. We certainly are in the boron doped diamond experiments.
Yeah, and I guess yeah, the overall effect is beneficial, as you've demonstrated. And the second part of that of Frankie's question is, has the potential difference at the bipolar electrode 1 volt, given that the biological electrolyte effectively would have a higher ionic strength, I guess?
Yeah. So one of the things that we don't fully the direct answer to the question is that even though the electrode is 1 centimeter long, and we're looking for a potential difference of 1 volt, and therefore one would expect that 1 volt per centimeter would be sufficient, we actually need to apply about 4 volts in that system to get a 1 volt potential difference on the bipolar electrode itself. So we're losing a significant fraction of the potential. We think it's important to try to map out where that electric field is being lost. We figure it's being lost close to the feeder electrodes. In other words, by the time you reach physically the bipolar electrode, the field strength is about 1 volt per centimeter. But we start out with 4 volts per centimeter. But as Frankie raises, the potential drops significantly close to the feeder electrodes because the electrolyte concentration is relatively high. So we lose a lot of the strength. And the important thing is that does not cause any biological issue, because where the biology is done, the electric field is still only 1 volt per centimeter and doesn't kill the cells, etc. The issue that it can cause is that you can now get unwanted reactions at the feeder electrodes themselves. And that's why ourselves and Gordon's group have spent quite a bit of time looking at different feeder electrode materials to make those feeder electrodes as inert as possible, because you don't want loads of water electrolysis. One of the systems we've looked at is actually forming oxide layers on aluminium so that they are highly passive towards any Faradaic electrochemistry. You want the feeder electrodes to be as inert as possible.
Thanks, Robert. I just got one more here, and from Andreas, and just about how homogeneous is the distribution of electric field across the wireless electrode width?
I think that's a good question.
Rather than length, I guess. Yeah.
And I suppose all we can speak to is mapping. So if we do a polypropyl deposition wirelessly, we can then obviously map not across the length but across the width, as you say. And that wedge-shaped film that we showed you, that's quite uniform to within 10 or 20% across the surface. So depending on the positioning of the bipolar electrode within the electric field, we would usually use quite large feeder electrodes compared to the size of the bipolar electrode, and that gives a reasonable uniform potential distribution. When we do the depositions, to round about 10% change in the sharpness of that wedge as you go across the surface. But imaging of the electric field distribution is certainly something that we will be ramping up.
Great. Okay, look, we might hang on. I'll just one last question here has just come in from Jerry Swigers. Considered have you, Robert, considered the shunt currents that may occur between the electrodes?
Sorry, if I can maybe just explain. In standard sort of electrochemical cells, industrial cells, they use bipolar electrodes all the time, and they set them up in such a way that there's no pathways for the current to go around the electrodes. So you get 100% transmission of the voltage to the next electrode. In your case, you've got the electrode sort of sitting in the middle of the electric field between the two sides. And so there's a large number of pathways around that electrode that'll create currents due to the electric field. They call them shunt currents. And that will be why you're having, I think, why you're having to apply 4 volts to get a 1 volt across your bipolar electrode. I just wondered if you'd considered that.
Thanks, Jerry. No, we had not considered that possibility, but I think your point is well taken, because irrespective of the electrolyte concentration that we use, even, you know, nominally electrolyte-free solutions, we will still need a higher voltage than we would expect on the basis of just the length of the bipolar electrode. So that's a really interesting point. Thank you.
Okay, we've got a few other questions, but I'm going to wrap it up there. I've certainly got a few other questions myself, and I'm sure Robert will be happy to take those offline by email or by other means of communication. But can I just thank everyone again for joining us today? I think you'll agree it's been an excellent presentation. I said at the start that Robert is held in high regard by his peers around the world. He must be held in higher regard than I even thought, because people in Ireland have actually got up to have a listen to this. And also, of course, from New Zealand, the UK, the US and India are all people registered. So congratulations, Robert. It was an excellent presentation. You've done some amazing things to date. And I think we would all agree that based on your trajectory that we're going to see amazing and exciting things for many years to come. So on behalf of the whole gang, huge thanks. And again, a big thank you for everybody who joined in, and let's keep the conversation going. So thank you, Robert.